Today we have a talk on overview of agricultural applications with SAR data. Okay, fine. Till yesterday's class, most of you have understood how the agricultural applications are done with the help of remote sensing data, particularly the optical data, the thermal data, and so on and so forth. So today we'll emphasize on what are the applications with this active microwave remote sensing part, particularly the SAR data. So in the first slide, you can see a few important crops which are grown in this rainy season. So this particular data set has an importance, particularly for crop monitoring in this monsoon or the rainy season, where you don't have sufficient numbers of cloud-free optical data available. As you can see, most of the part of India right now, it is overcast. The sky is overcasted with a lot of clouds, mostly entire part of the day and continuously for months together. In right side, you are seeing the picture of our own Indian RISAT satellite, which I will discuss in detail. So these are some of the crops. No, you can see the kharif crops mostly, and mustard is a rabi crop. So lot of applications, lot of studies are being carried out with the SAR data. So the electromagnetic spectrum you have studied in your earlier classes consists of a large spectrum of wavelength. So the visible light, if you can see here, my pointer, the visible domain is a very small part. So our eyes can sense mostly in this domain, and the optical remote sensing basically makes use of this particular wavelength only this fraction of the entire spectrum. So the microwave radar wavelength is located here. You can see it is much longer wavelength. Henceforth, it can penetrate through the clouds. Even some of the wavelengths can penetrate the rain also. If you have moderate intensity of rain, it can be penetrated. The radar initially was mostly used during the World War times, and a little bit on the history just to make you acquainted. Okay, so earlier how the remote sensing evolved, the radar remote sensing, how it evolved is actually it was used during the World War time, and basically these are some of the codes. You can see the wavelength Ka, K, Q, Ku, X, C. These are some of the codes, and they have a particular wavelength and frequency associated with these particular letters. So some of these domains, especially starting from the X, C, S, L, P, these ranges, these frequency ranges are used in remote sensing in the present days. And the other part, okay, the other part of the spectra where you have the high frequency waves, they are not much used in remote sensing. Those spectrometers and other sensors, other active sensors use them. Okay, but remote sensing mostly uses this X, C, L nowadays. And IEEE has some convention. What is the microwave region basically? So it is 300 megahertz to 100 gigahertz. Okay, so this is the wavelength range or frequency range where it operates. In the right hand side of the slide, again, you can see the different wavelength. Mostly in today's context, C band is very widely used for agricultural applications. So it has a wavelength of around 5 centimeter. The mean is around 5 centimeter associated. So the entire spectra is from 3.5 to 7.5 roughly. And then towards the longer wavelength, we have S, L, and P. So maximum it goes up to 100 centimeters or so. And then towards the shorter wavelength, we have X, Ku, K, Ka, and so on. Now, how these wavelengths are decided, what kind of application can be done with what wavelength, what frequency, those things we'll just see in our subsequent slides. Now, few questions may come to your mind at the beginning of this class, that what are the advantages actually? Why we should go for microwave remote sensing? First of all, I told you that cloud cover is a major issue, particularly in this crop season, the kharif season from June to September. Cloud-free optical data is rarely available, or it is very minimal. So none of the day is totally cloud-free. And also day and night coverage, if it is required, this is possible with again microwave remote sensing. Above these two advantages, it also provides a complementary information to the optical data. It's not that optical data is good or SAR data is good or which one is better. The question is not about that. One complements the other. Actually, what we sense from the optical data is mostly the chlorophyll related properties, the pigment related properties at a very micro scale. But from SAR, we can sense the macro scale properties, okay, the moisture, the geometry, and so on and so forth. So they complement each other. Major disadvantage of this data set is that they're difficult to interpret. Not so difficult, but we'll see how to interpret that. Okay, there may be some differences from optical data, and also the coarse resolution except SAR. For other microwave remote sensing sensors, the resolution is a bit problem, but for SAR, we have high resolution datasets available. Now coming to the passive and active system, most of the optical remote sensing, the thermal remote sensing, mostly whatever you have studied till now, until yesterday's class, they are mostly passive system. Okay, they use sun's energy. But in the active system, the instrument pulse, it needs its own power to operate. It is not dependent always on the sun's energy. So it creates its own electromagnetic radiation that is transmitted from the sensor towards the terrain. Then it interacts with the terrain actually. The whatever energy is transmitted, it gets interacted in a different manner. So it may be scattered, may be absorbed, may be transmitted. So a part of the scattering, okay, this part of the scattering which is sensed back to the sensor, so that is known as the backscatter and is recorded by the receiver. So this is the entire process which goes into play in the active remote sensing part. Similarly, there is passive sensing in the microwave domain also. Like your optical domain has mostly the passive sensing, here also we can have passive sensing. So it is based on the surface emission, the background, the rain emission, and so on. The concept of brightness temperature, what you might have already heard, you can see the right hand side diagram. So surface temperature and emissivity, it gives rise to the brightness temperature which is sensed, and there is a relationship between the brightness temperature and the kinetic temperature of a body. So the kinetic temperature is the temperature which you are sensing through a thermometer, and then it is related through emissivity. Okay, so these are some of the key points on active and passive sensing. So active microwave sensing, mostly there are large number of applications in different domains apart from agriculture, and scatterometer is one. So the principle is same as radar, but the scatterometer operates at little different frequency. Mostly Ku band, Ka band, even C band scatterometers are also available. So they have agricultural applications also. But the image formation may not be as sharp as in case of your SAR data. Then there are side-looking radars. The disadvantage of vertically downward-looking radar is that their ambiguities have to be overcome. So the radars are mostly side-looking. In case of optical data, mostly you see they are nothing-looking. Okay, so we don't have this ambiguity problem. But in SAR data, because the transmitted pulse is to be recorded, it interacts with the target, it is recorded. So the left-right ambiguity is a major issue. So to avoid that, in order to avoid that, we go for a side-looking system. So there may be RAR, that is real aperture radar, SLAR, okay. And the time delay actually is measured, the distance from the antenna, the backscattered intensity, which gives rise to the terrain characteristics actually. What backscatter is actually sensed in the receiver. So whether it is low, high, medium, so that gives us an indication how the terrain is actually. So what all properties of the terrain affect the backscatter, those things we are going to discuss in detail. Now, based on the disadvantage of the side-looking radar, the SAR was designed. So basically the resolution, initially the systems were having very low or the coarse resolution, and then SAR was developed keeping in mind this, and resolution was improved. Then the Doppler effect, the frequency shift, and those things, they play important role in formation of these data sets. And the concept of radar equation, if you see the right hand side diagram, a power is transmitted, and then there are scatterers, the target, it interacts, and then power unit area at the receiver. Okay, so these interplay takes place, and then what this equation you were seeing is, it is known as the important radar equation. This is important when you are actually trying to compute the backscatter, understand the concept. So this equation may be important. But when you have the data set, when you have the SAR data, You need not derive this. We have the backscatter already computed in some form. Very simple equations, not this equation. This need not be applied. So how you have to process the data, those things also will see in the coming slides. As I was telling you, the radar backscatter is dependent on macro structures, macro scale properties of the target. So here we are dealing with the vegetation. So the dielectric properties of the vegetation of the soil. The dielectric is a direct measure of the moisture content of the medium. Then the geometry, the shape, the size, and again the wavelength of the radar, how much it is able to penetrate, that will depend on its wavelength. So again, what kind of geometry, shape, how much can be studied, how much it is able to sense, all these things will depend on the wavelength. So some important properties, okay, there will be sensor properties as well as the target properties. Sensor properties like incidence angle. Okay. So at what incidence angle you are acquiring the data, then at what polarization you are acquiring, at what frequency you are acquiring. These all are important sensor parameters which has to be taken care before you are actually ordering the data. So you must understand at what values of these parameters you need the data. So that again depends on your application. Suppose you are going to do a soil moisture application, or you are going to do a vegetation parameter retrieval application. So the data configuration basically will differ based on your need. Then the roughness. Now coming to the target parameters. What will be the important target parameters? There will be roughness, the moisture. Basically these two are the important ones. So roughness will depend on the geometry, the shape, the architecture of the canopy, and the moisture content will again depend on the condition of the crop, suppose the condition of the vegetation, the nature of your stress if it is there, or whether it is properly amply irrigated. Those things will determine the moisture content. This volume scattering is basically, it is a little different term. These terms are not used in your optical sensing. So whatever volume is there, the canopy volume, they give rise to multiple scattering actually. And that can be directly sensed as backscatter. So for soil, what frequency? I was telling you the configuration. So for soil moisture estimation, lower frequency radars are best suited. So they are able to penetrate more. They have to penetrate and reach the soil in order to sense it. If they get trapped or they get lost in the vegetation medium, so our purpose is not happening then. So they have to travel deeper inside the soil, deeper into the, at least reach the soil. Whether they will travel into the depth of the soil or not, again depends on the moisture content of the soil medium itself. Then the crop information specifically is possible at higher frequency like Ku band, X band or C band. So at higher frequency, the penetration, the wavelength will be little less than the lower frequency. So it may be Ku band or X band or C band. So it need not penetrate the entire canopy. Suppose we want the information of the top layer of the canopy or up to a particular depth of a wheat canopy or rice canopy. So we want the wavelength to be penetrate only to that particular range. It should not go and get into the soil. Okay, so that we get the vegetation information from the upper layers. So Ku band, X band, C and L band are important for crop type mapping. Now explaining what I was telling you about the volume scattering and surface scattering. Just see the first diagram, top first diagram. Okay, so this is the trunk and this is the canopy layer. If you can see the first dotted line. So this is the entire canopy from here to here. Okay, so there will be some different type of scattering what we can sense through this SAR remote sensing. So there will be the trunk and the surface. This is the surface of the soil, and this till here is the trunk, and then you have the vegetation canopy. So directly it will be reflecting like this. Okay, so this is known as the double bound scattering. Then some signal will directly penetrate like the L band, no, for soil moisture sensing. It will directly penetrate and then again it will come back. So this is the surface scattering. Some will interact with the vegetation medium. So there will be multiple reflections. So this is volume scattering. And then some will interact with the surface volume. So multiple scattering from surface and volume. Okay. So these are some different types of scattering which takes place in the vegetation soil medium. Now we were discussing about the incidence angle. So some important concept, if you can see, this is the Earth, this right hand side small diagram where you have a blue Earth. So this is the incident radiation. This is the incident ray. Okay, the ray which is coming from the radar and it is reaching the ground. And we have drawn a perpendicular from the Earth. So the angle between this perpendicular and the radar line of sight, this is known as the incidence angle. So this incidence angle is also very important. Like it is an important sensor property which governs what kind of backscatter responses we get actually. So higher the incidence angle, it will sense mostly the top. The lower or the narrower the incidence angle, it will penetrate deeper into the middle. The next diagram, just below that, if you can see, this is a little undulated terrain is shown here. Again, the same concept. You have the radar line of sight here, the extreme right hand line, the thick black line. If you see, this is the radar line of sight, and then we have a perpendicular. This is the vertical from this surface. Okay, the red dot, from where the red dot is there. So this angle theta from radar line of sight to the vertical. So this is the incidence angle. Now, if you have a surface normal, the surface is like this. So if you draw a normal from there, this theta 1, whatever you are able to see, this is known as the local incidence angle because it is taking the local terrain characteristics into consideration. These details are explained again in this left-hand side image. Here you have the altitude where the sensor is operating, flying. It is kept at this altitude. So this is called the range direction and this is the azimuth direction. So this is along track and this is the across track. And you have again the same concept, the incidence angle. Here is the incidence, you see the radar line of sight, and the vertical, this is the incidence angle. Okay. And similarly, 90 minus that, you get the depression. So again, the local incidence angle, this far away from this point, this is where you touch the ground. This is called the far range, and the nearby point near the ground, this is what you touch is called is the near range. So this gives you the entire range. Swath is got from the difference of this near range and the far range. Now coming to, these were some of the background just to see how the data acquisition occurs. Now coming to the SAR systems, and then we'll finally go into the agricultural applications. Frequent cloud cover actually is a hindrance during the monsoon season. Already I discussed this, and also in winter season also, especially in the northern India and few parts of the southern India where the winter monsoon is there. Again, this problem persists in the winter season also. So because SAR is self-illuminating, it has all weather day and night acquisition capability for the data. And hence, this is an advantage of this system. So current spaceborne SAR systems are available in 3 frequencies, that is the C-band, L-band and X-band. For agricultural crop monitoring, mostly the C-band SAR is found to be most suitable. A variety of beam modes are available from all these sensors. So some of the beam modes and some of the applications of the RISAT satellite we'll discuss in more detail. Operational C-band SAR system currently available are RadarSat-2, Sentinel-1, and recently our own RISAT-1A, that is the radar imaging satellite. It is a follow-on mission of the first satellite which was launched in 2012, and the current one is launched in current year during the month of February. So RISAT-1 MRS mode, the first satellite, it was operational till 2016 and around 36-degree incidence angle. Okay, so this is little wide incidence angle and swath of 115 kilometers and pixel spacing of 18 meters. So this is the standard systematic mode which was acquired by this satellite. And the follow-on launch happened during mid of Feb, RISAT-1A, and now it is operational. We are getting the data of this satellite right now. So for crop monitoring, as you understand from optical data, what you have studied, that you need multiple wavelength bands to compute the various indices like NIR band, IR band, green, whatever. But in SAR, you know that this is only one frequency. So you will get black and white images, right? If you have only one frequency, you don't have multiple wavelengths. So you get one image as a black and white image. So how to derive information out of that? Generally, we take a multi-temporal data set. So we acquire the data on subsequent dates to derive a meaningful information out of that. Now, these are some of the modes which is available of the RISAT-1A, this EOS-4 satellite. If you can see, there are fine resolution modes, medium resolution mode, and the coarse resolution mode. FRS-1 and FRS-2 are the fine resolution strip map mode. Okay, MRS is the ScanSAR mode, CRS is also the ScanSAR mode. So you have quite high resolution. In the FRS mode, you have 3 meter into 2 meter and 3 meter into 4 meter. In the MRS mode, you have 33 into 8. These are range resolution, azimuth and range resolutions are given. So you have two figures in this. And when you make when it is ground projected, you get a square pixel. So we have a pixel spacing which is square. Okay. So what is the worst sigma naught which can be sensed? So it is around -18, -19 dB for all the modes. And swath is quite high. Swath, if you see, it is in few hundreds of kilometers for this MRS and CRS mode, 160 kilometer and 220 kilometers. But for FRS, we have a smaller swath. Okay. This is how this is our own site in NRSC's own AD site. If you visit, you will see the entire mosaic is available with this data set, the coverage of the US. So depending on your requirement, you can order at what incidence angle, at what polarization you need this data, it is available. And another important contribution of this RISAT-1 satellite is that the circular or the compact polarimetric mode was first developed in this satellite in 2012, and in the follow-on mission, in the current mission also, it has the compact mode as well as the fully polarimetric mode, where you have all the four polarizations combination like HH, HB, BB, BH, and the phase is preserved between them. So you have all this information for your analysis and applications. These are some of the global sensors in the microwave range. I'll not discuss in detail, but you have so many sensors. Some are active, some are passive. It is enlisted here. So radar set, as I was mentioning, is mentioned here. Cosmos Skymet, IIRS, these are all important sensors which has been tremendously used for its agricultural potential applications. Okay. Now, crop discrimination. Let us come into this part because our focus will be on agricultural applications. So the backscatter response is function of these factors. We already discussed the crop roughness, the moisture, as well as the geometry or the architecture. Various crops have different backscattering properties based on their canopy structure. You know that rice crop is different from mustard crop, is different from cotton, is different from jute. So they will have their own canopy structure, architecture, as well as the moisture content, which governs these backscattering properties. In rice crop, mostly the transplanted rice, which is grown in this particular monsoon season, the background standing water has a significant contribution. Okay, so it has a specular reflection in the initial and the major part of the growing season. Okay, so very low backscatter is observed in the initial season, but as the crop grows, it picks up. So those details we'll see. Where there is no background water, like in case of the other crops. So other important factors like orientation, density, canopy, soil moisture, those things play a role in governing the backscatter response. So before going into the data and analysis part, little bit of processing, how to process. So I thought I just will spend couple of minutes on this. So various softwares which you can use for processing the SAR data is listed here, and just have a look. And the Sentinel GRD data you can download from this Copernicus site. Also, Google Earth engine can be used to process the data right now. Nowadays, since a couple of years, so it has all facility without downloading the data itself. You can process and do your analysis very seamlessly. So these are some of the important steps which you need to do if you have already downloaded the data. So these steps you can carry out in the SNAP software. Apply orbit file correction, do the calibration, then speckle filtering, terrain correction, and if you have to use multiple dates of data, then go for co-registration and convert their data into decimal. So these are the some of the how it looks. Just a view of the software for the processing platform. This is the SNAP developed by ESA, European Space Agency. The Sentinel. This is the Sentinel data platform, and this is how your black and white one single date, single frequency image looks like. When you process after speckle filtering, speckles are some salt paper noise, which is inherent characteristic of the SAR datasets. So they can be, it results from the interference of the signal, the coherent signal, and it can be removed by some processing, especially the adaptive, using the adaptive filters. So after speckle filtering, you have more smoothened image. The salt paper noise will lessen in magnitude. So it is, the data is more prone to applications. Otherwise, with those speckle noise, it is difficult to sometimes interpret the data. So it is important, and it is a necessary step to remove the speckles, minimize it at least. Okay, so this is how the processing goes, and then for classification, you have some of the tools available here. Otherwise, after processing, after creating the multi-temporal set, you can take it elsewhere for analysis. Okay. So now coming, like, you have various crop types, the geometry, the architecture of the crops, it may be broad-leaf, narrow-leaf. So increased backscatter is resultant of the biomass and the water content. So there are tonal differences, high level of backscatter at C band or X band. So they result in, manifest in terms of the levels of backscatter. Also the phenology, when you are taking temporal data, the crop is growing, as I was mentioning. So when rice, it grows from the background water after transplantation due to more number of tillers, more number of shoot growth, it will have increasing scattering, increasing volume scattering, and this will be again manifested in terms of increase in the backscatter. So what are the plant parameters what are important when you go to the field? These parameters are to be measured, and they can be modeled actually to study the crop growth and dynamics. So the plant height, the stem diameter, stem density, leaf area index, leaf length, width, thickness, various leaf related parameters, then plant biomass, plant water content. Okay, and then there are various inversion algorithms available to estimate these parameters. Some of the old examples I'm just quoting, but there are a lot of papers, lot of literature available in today's context, which you can go through. So what is the optimal data? This question might be arising in your mind right now, that we have to acquire the data. When should we go for the data acquisition? So the first acquisition, I'm taking a simplistic case for rice crop. The first acquisition will be sometimes coinciding with the transplantation stage. So it may not be the exact date of transplantation. It may be 2-3 days here and there, doesn't matter. Okay, but not much. So that you are able to sense, or you are able to monitor the crop growth in a very, very efficient manner. The second acquisition will be roughly around the tillering stage. Then, so it may be around 20-25 days after the first date. Then the third date will be around the peak vegetative stage. So it is around another 24-25 days after the second acquisition. And then finally, if you want a fourth date, it can be during the flowering stage. So operationally, what is being done currently is there are 2 forecasts which are given by the Ministry of Agriculture. Now this work is operationally done by Mahalanobis National Crop Forecast Center, located under Ministry of Agriculture in New Delhi. And they give this forecast twice. Okay, so 45 days after transplanting, the first forecast goes, which accounts for about 75% of the season's rice. And then the final forecast goes before the 30 days before harvesting. So by adding another date after 24 days from this 45 days. So this operational methodology has been developed by ISRO, by our team, and now it is being carried out by various ministries also for their states. Now, how the multi-temporal data looks like. So a single data of the SAR will look black and white. If you see here, there are 3 images, 3 black and white images. The first one is during mid of July. The second one is during August, and the third one is during September. So I think you can't infer much from these three data sets, but when you combine them, they actually give you a very meaningful information. So what is happening, if you see these dark patches in the first date, most of you can see this. So the first date during the July, you can see mostly the image is bright, except few patches which you have this dark tone in the central southern side and little bit in this central middle part. Okay. So what is happening? These fields are dark because they are having specular reflection. The backscatter is low due to standing water due to transplantation of the rice crop. The next date during the August, what is happening? Most of the part of the image is dark. So most of the part is getting transplanted during first week or second week of August. In the third date, what is happening? Only few patches again, very small area is getting transplanted. Otherwise, the entire image has become bright. How? Because these fields which were transplanted during the month of August, now the rice crop has grown. And due to growth of the crop, the backscatter has increased. So this bright response is due to that, due to multiple scattering, volume scattering, which have been explained. Now, because you have these 3 dates separately, so in order to infer what we have the 3 colored guns, the RGB, no, where you put in optical data, what you put the different bands, green, red, and NIR. Mostly you put that. Here what we do, we put again in RGB, we put the first date, second date, and the third date. Here first date is July, second date is August, and third date is September. So we load them in R, G, and B. What will happen? Now, if we have a high backscatter on the first date, it will appear as red tone. Okay. If we have high backscatter on the second date, it will appear as green tone because it is in G, green band. And similarly for blue. So if you see this colored triangle now, if we have high backscatter in the first date, we have red. If it is in the second date, we have green. And if we have in the third date, it will be blue. But suppose there is a low backscatter in the first date, and there will be high backscatter in the second and third date. Suppose you imagine these points where you have low backscatter in the first date, and few of those fields have grown, okay, in the second date. So you'll have high backscatter in G and B. So you'll have a cyan tone. So high backscatter on second and third date. This particular tone you'll get in the image. I hope everyone is understanding this concept. Okay. Now suppose if you have a low backscatter in the second date. So consequently, there will be high backscatter in the first date and high backscatter in the third date, but low backscatter in the second date. So you'll get a magenta tone. Okay. Right? High backscatter on first and high backscatter on the third. So combination of red and blue will give you magenta tone. Similarly, if you have a low backscatter in the third date, so low backscatter in the third date means high backscatter in the first and second date. So some of these points, no, here you have third date low backscatter, but corresponding here you have high and here also you have high. So these are very late transplanted rice where you have transplantation only very late in the September month. So those points will appear as yellow when you have a combined color image. So this resultant, what we have been discussing right now, the image will appear something like this. When you combine RGB of those 3 black and white images, it will look something like this. So this cyan color, what is written here as early rice. Okay. So this is early transplanted rice, and most of the images it is occupied by the magenta patch. So this is late rice. The transplantation occurred during August. Okay. And the very late part where transplantation is occurring during September. So it is yellowish or greenish yellow in color. So this is how the SAR image now looks, very colorful when you combine the 3 dates, and it has all sorts of color, right? Like you used to see in the optical images, colorful images, here also you can, and you can derive meaningful information from this combination of the 3 dates. So this is how your crop is growing. The blue line, if you see, this particular second date where you have low backscatter around -20, -18 dB. So this corresponds to the transplantation stage, and then as the crop grows, the backscatter will increase with the tillering, then vegetative, and so on and so forth. Vis-a-vis, if you see the water bodies, no, the permanent water bodies, either the river or the other features, waterlogged area, they will have a low backscatter throughout. So this magenta line, if you see, all the dates, these are the various dates of acquisition, all the dates, they will have a low backscatter. Contrary, urban areas will have high backscatter in all the dates due to dihedral scattering. So this yellow triangle jointed lines, this is for the urban patch. Intermediate will be the crops. Okay, they will neither have that low nor very high backscatter. So for other crops like cotton, maize, you will have initially comparatively high backscatter than paddy. Paddy has very low initial backscatter due to the standing water. Okay, so other crops may not have standing water. They will have high soil moisture during the sowing period, and the field preparation will result in high roughness also. So they will have a relatively higher backscatter. And then with the crop growth, then there will be smoothing, and then there will be crop growth. This will again lead to increase in backscatter to some extent by around 3-4 dB, and then it will stabilize and it will fall again with the harvest. So this is the trend you see for maize, cotton, sunflower, and other crops which do not have standing water background. So suppose you want to go for a classification scheme based on this logic, whatever we have studied till now. So we delineate the phenology, crop phenology based on the ground info. We take multiple areas where soil conditions similar. So this will help us in relating the backscatter with the canopy conditions. And then we generate a knowledge base. We create a decision rule, and then we go for classification. Just I'm showing you the results, how we do that. So this left hand side, whatever you are seeing, and this is a part of that same FCC what I have shown you earlier. So now first what we are doing, we are classifying the water body. So if all the dates you have values less than -17 or -18 dB, so this has picked up the water body. All right. Next we are taking the urban. All dates you have values more than -3 dB, all dates. So these pink patches have got picked up, and water already is classified in the last step. Next we go for fallow. So fallow, what is that fallow? It's not the rice crop, but the other areas where you may have some grass growth, or it may be non-vegetated fallow. So the backscatter will lie between -12 to -5 dB in all dates. So these pink areas near the river beds, these are some of the fallow areas, and also in the agricultural area you may pick up some point. Now coming to the rice crop. If you focus on the normal sown rice crop, it will have a low backscatter in the first date coinciding with your transplantation, maybe -16, -17 dB to -12, -13 in the first date. Then in the second date, there will be a 2 dB rise. Then in the third date, subsequently, another 2 to 3 dB rise. So this is the normal sown rice crop. In the late sown crop, what will happen? Your first date will be high backscatter, but the transplantation happens in the second date. So second date will have a dip. So that may be -18, -15 dB in the second date, and then subsequently the third date again it will pick up. So this small green patch, what you are seeing in the southern part of the image, is the late crop. So now this is the classification, and based on the sample segment, the sample segment is sometimes practiced for few crops, and for other crops it's not practiced also. But it is better for a like major crop when you are doing acreage of rice or some major crop like cotton. This sample segment approach is always advisable, because it will not go into classifying the non-agricultural areas. Okay, sometimes in order to avoid these errors, this sample segment approach is necessary. So these are basically 5 by 5 kilometer grids, what these squares you are seeing here. And based on the ground truth, it is done to accommodate the difference in management practices. So there may be different categories of agricultural sample, the high proportion, the medium proportion, the low proportion, so on and so forth. So for example, the total segments, we have taken an example of a particular district in West Bengal. The total segments is around 552, and out of that crop segment is 533. And the sample segments, the small end, the sample population is 72. So crop proportion is basically what? Rice pixels by the total geographical pixel. So crop area will be crop proportion multiplied by the number of segments into the segment area. So likewise, we compute the crop area. And then we go for the acreage estimation. These things you might have seen in the optical data. So how we go for do for microwave data that and the SAR data that we are discussing right now. So crop proportion per segment is calculated. Then the proportion is multiplied by the capital N, that is total crop segment, to get the crop area for that particular stratum. Then we have to correct for the pseudostratum for any like the unsampled points or unmeasured points, and based on a factor derived from the geographical area to n segments area. So this is corrected for the pseudostratum. The aggregate the stratum wise figures. So there may be different stratum based on the different proportion of the crop growth. So all these segments may not have equal proportion of crop. So that is why the stratum concept is there, and then it is finally projected to the national rice area. Okay, so this is how the acreage estimation is done. Now, this particular figure actually is the application of this. Once you compute the rice area, this green part is the state of Orissa. All the districts are shown using these yellow borders, and the rice area is seen as the green layer. And this cyan layer, the blue layer with what is overlapping, is the flood layer. So there was a cyclone in 2013. There are a lot of damage happened to the otherwise the infrastructures and some part of the rice area also. So this monitoring was being carried out using RISAT and RadarSat data for flood area estimation. So both cross and co-polarized response was monitored, and different dates of data which were acquired are shown here. Okay. So whatever area has been flooded and whatever area was damaged, the crop damage assessment was carried out, and such a project or such evaluation can be done anywhere. In the every year, floods are occurring in different states of India like Assam, Bihar, and so on and so forth. So this kind of damage assessment study can be carried. So next, using both the data set, the SAR as well as optical data sets, I already mentioned to you the different advantages. No, they are complementary to each other. So if you can see here, this top is the SAR image and the bottom is the optical image. So conjunctively, they can be used for either stress or crop condition monitoring or crop health assessment. So RISAT data and IIRS data has been together used in the past. This is the old research. So such kind of synergistic study in other crops as well as in rice crop is there. And this has a way ahead because conjunctive use, complementary use of these data sets is very, very important. Now for crop type identification, crop biophysical parameter estimation, what kind of data do we require? Multi-temporal data is very important, that we have seen in our past slides, how meaningful information is derived from multi-temporal data. Then multi-frequency. Okay. Temporal means you need to pick up the data sets corresponding or coinciding with the important critical stages. Okay. And it may be 3 date, it may be 4 date or 5 date and so on. And almost correct classification with very high accuracy is possible when you are able to identify these critical stages. Then come to the multi-frequency concept, where you have various layers in the canopy. Suppose if you have very high biomass vegetation, so the C band may not be able to penetrate. So you can use multi-frequency. You can combine C band and L band or X band and C band to get differential crop information of the different layers. So for example, forest studies, you go for L band and T band. For crop studies, you can go for X band and K band. So depending on the biomass, depending on the grain crops or non-grain crops, we can decide the frequency. Then sensor, multi-sensor, like combination of optical and microwave data together can give us some advantage. Then multi-feature, combining tone and texture discriminants from a single image. Textual information useful for forest species, for example. Then coming to the biophysical parameter studies. We have already listed some of the biophysical parameters which are important both from optical as well as SAR sensor point of view. So this is your height, plant height, density, LAR, biomass, so on and so forth. If you see this left-hand side graph, these are simply plant height variation with the backscatter. So as the crop grows, the height increases as well, which in turn manifest in terms of increase in the backscatter. So we have shown this in two frequencies, L band and C band. You can see C band, this orange one and the cyan one, these are the C band, and this magenta and the green, this is the L band response. So the response is more stronger for L band at the later stage of the growth, that is in October, as compared to C band, the cyan one. But the response is better for C band in the initial part of the crop growth phase. So in the initial phase, when the biomass is less, C band serves our purpose. But for at a later stage where your biomass is more, you need more penetration, then we have to use the longer wavelength, for example, the L band. Similarly, this is for L band HB and HH. So the cross polarization response is more strong at higher plant height as compared to the co-polarization response. Okay, due to the multiple scattering interaction. And the right hand side, these figures, just to give you an idea of how the biophysical parameter varies with the crop calendar, basically. So height, if you see this red line, it is increasing, but it will remain stable. But if you consider the LAR, the leaf area index, it will increase, but then it will have a peak and it will decrease much before harvest. Okay. Then the stem density will also increase, then it will slightly decrease and stabilize. So these parameters we need to measure in the field, and we can relate it to the various SAR parameters. Then similarly for fresh weight, leaf fresh weight, and other water content, the water moisture content, the total fresh weight, these all will increase as the crop grows, and they will have a definite relationships with the SAR parameters. For example, rice biomass exercise has been carried out by our team for all the important rice growing states, and this is also being operationally carried out by Mahalanobis Crop Focus Center located in New Delhi under the Ministry of Agriculture. So the biomass and the transplantation date retrieval from multi-temporal ISRO data, this is being done with a very high accuracy, and the transplantation date can be retrieved with an accuracy of ±2 days. So this particular graph, whatever you are seeing in the left-hand side, this is how the biomass is varying. This green triangle curve, you see how the biomass is varying. And this purple curve shows you the frequency plot. So there are two peaks. You see, this is the x-axis, whatever we are showing is 0 to 120. This is days in the Kharif calendar. So first means June 1 until it goes till October. So there will be peak at 30. 30 means end of June, and then another peak at around 50. So that is 20th of July. So these two peaks represent two peaks of the transplantation. Okay. And then how the biomass is changing, biomass is increasing. So biomass map is produced, whatever you are seeing in the right hand side. We have high biomass in this orange area of 4 kg per meter square or more. Then yellow and green and cyan are the lower values. An application of this biomass map of rice, this was carried out in the Bringing Green Revolution in Eastern India. So the training was imparted to MNCFC, as I have been telling you, and this higher wavelength data will have more potential. So using this tool of biomass and transplantation, we could monitor this project, and higher yield by about 30% were found to be reported using this multi-temporal ISRO data, ISRO MRS data. This map of Orissa, you are seeing multiple sampling points, and so this particular program was a great success. Okay. Another important crop is a jute crop. The lower images, whatever you are seeing. So Nagaon is a state of Assam where jute distribution from 3-day 12 polarized ISRO data was carried out during 2016-17, and this input was given to the Ministry of Textile. Here, if you see these two maps, the greenish one, so this is a plant height map, because jute crop, you know, this is an important fiber crop grown in 2 or 3 states in India and also in the country of Bangladesh. So the yield is the fiber, so it is directly proportional to the plant height. So if you are able to map the plant height, you can directly relate it to the yield of the crop. And also the vigor. This magenta and cyan map are showing you the vigor. The cyan is the low vigor, magenta is the high vigor crop. So these outputs can help giving inventory and monitoring the crop, how they are growing and how the yield can be related. Now, coming to we have been talking about polarization in the beginning of our lecture. So few minutes I'll dwell on the multi-polarization applications. So if you see this particular figure, you can see this only one particular image, one particular date image. The left hand side is of October and then right hand side of December. You might be wondering how they're so colorful. Okay, so this is the compact polarimetry data, what we were discussing for ISAT. This is the output of that data, the M delta decomposed image. So basically we are having various types of scattering. We can actually partition these scattering into surface, double bounce and volume scattering. There are various equations in which enables us to do so. And the surface scattering is mostly represented by this blue color. So the blue, the green is the volume scattering and the red is the double bounce scattering, which we are able to see in this urban area. So this particular data set is Hisar and its surrounding area. These big plots, crop fields you are seeing, these are the part of the Central State Farm in Hisar, Haryana. And now we'll come to interpretation of these colors actually. If you see this in this magenta circle, this blue box, okay? So these are the mustard crop, which is sown during September, October. So it has high surface scattering during the initial date. And when the crop grows, it will transform to volume scattering. So it has grown and in December, you see this same field, this square, it has turned into more of cyan color. So the volume component has also come into significant amount in this. The surface component has decreased here. Now, if you consider the other patches, like these are also mustard patches. Here you see entire blue, but here it has become greenish due to more volume component. The cotton crop, which were already the picking would have been carried out and during the October month, and it is already in the volume scattering stage during September, October. Now during December, most of the fields are harvested and there will be mostly surface scattering. So it will again have dominant surface scattering during December. So such kind of interpretation is possible from a single date image from this polarimetric data set. So this is a compact polarimetric, it's a partial polarimetric data set. From full polarimetric, even more information can be derived. And bottom here you can see some of the plantations are there, these patches. You're able to see my pointer. Sorry. So these green patches, these are plantation which are there in both the dates. So these are some orchards and some plantations. Now in wheat fields are seen here. If you see these blue fields, so in October, November, mostly they are fallow, and in November the sowing has started, and then they will mature to some extent 1 month, 1.5 month crop in December. So the fields are, some amount of volume scattering is seen in those fields which were totally surface during the October month. Another example, this is part of Gujarat and this is 2015 data. So in the north, this is a river, you can see the bridge very clearly. So this particular image, left hand image is of January and right hand image is of February, just after 1 month. So this cyan northern patch, this is the orchard, and this reddish patch, whatever you are seeing, the magenta patch, this is due to double bounce scattering, as I was mentioning. So wheat crop, this is late sown wheat crop, is having double bounce scattering. The other fields are already in the volume scattering, if you can see in the southern part, which is slowly transforming to surface scattering during February. In Gujarat, you know, the summers are very early and the harvest is already done for the wheat crop during February. So these things are picked up very prominently, very clearly using this polarimetric data set. This is another full polarization data set from ISAT, and multiple colors are actually showing the different crops. The wheat, mustard, gram, this is a winter image during December. So this is a classification done from the same data set. With a single date, you can do the classification of various crops using some rules of the various polarization. You can give a threshold, or you can go for various decompositions and then various parameters. So those things, what decompositions and all for full pole data we'll discuss. So this one is like you can derive the entropy related, the eigenvector, eigenvalue related parameters, that is entropy, anisotropy, alpha, and so on. You can see different crops are having different responses. Okay. Based on these scattering components has been carried out. Then another index is there, like in optical remote sensing, you have heard about simple vegetation index, simple ratio index, normalized difference vegetation index. In case of SAR, you have few indices like the radar vegetation index, which is actually manifesting the scattering in the HV and the HH and the VV, all the four polarization components. So more the crop growth, more the vigor, more will be the RBI, more the water content. So what you can see, this exercise was done for cotton crop. These green images, whatever you are seeing. The left hand side, the red box, you can see if you the red oval shape what is being shown here, few fields are brown and the next date it is turning to green. The crop is growing, so the radar vegetation index is actually increasing. Similarly for this blue oval also. The temporal radar vegetation index in Surendranagar, this is important cotton growing district in Gujarat. Okay, so red circle represent the PT cotton dominated area, and the blue circle basically the indigenous cotton. So indigenous cotton is actually very slow in growth. PT cotton is very fast, so the difference can also be seen in terms of the radar vegetation index. And various parameters were related to this index, like the water content, the vegetation water content, the biomass, etc. And those maps have been prepared from the single index with a very high accuracy. Now, if you see this left-hand side images, the radar vegetation index has been computed for the winter crops, mustard and wheat, you can see. So low biomass versus medium and high biomass, how the response is. So this is a very sensitive index and it varies from 0 to 1 again, like your other indices, and the values can be little higher than 1 also when you use the polarimetric RBI, so it can be up to 1.3 basically. Various decomposition methods, various decomposition components can be actually computed based on the fully polarimetric data set. So this is your HA alpha decomposition, again eigenvalue, eigenbased. So these are the various zones Z1 to Z9. You can see in the left hand image. So this is the plot of alpha angle versus entropy. These two important parameters, the alpha angle gives the dominant scattering angle. So it varies from 0 to 90. 0 to 30 is mostly the surface scattering, 45, 50, 60, this is mostly the volume scattering, and near 90, 70 to 90, this is mostly the dihedral scattering. So the buildings, the urban structures will mostly fall in this Z1, and the crops will mostly fall in this Z5 and Z6. So high biomass crop will be more towards the Z5, and the low biomass crop will be towards Z6. So this discrimination, this was possible for various crop types. Some mixing is there, but with some other parameters like anisotropy, this discriminations were possible. Then another important parameter which can be derived from partially dual polarimetric Sentinel-1 data is the entropy, the Shannon entropy, which was again related to the various crop parameters. As you can see, the crop height, crop biomass. So moderate sensitivity was found in contrary to a very high sensitivity from the full pole data. Moderate sensitivity was found, but still the crop sowing calendar differences, those things could be picked out. And even the stages, the phenological stages could be mapped. This left-hand side bottom image, you can see various dates of the winter images for the central state fund. We could sense the various stages of the mustard and the wheat crop. The same study with the different machine learning models, these various stages could be actually retrieved. The stages of the phenological stages of the wheat and mustard crop. Now this is another concept of polarimetric signature, where we plot the orientation and ellipticity angle and we measure the normalized scattering. So this is the tau and the phi, the ellipticity and the orientation angle. And here what you are seeing in the z-axis is the normalized scattering. So for settlement, and we have the co-polarized and the cross-polarized response. And for different crops, you have a different kind of polarimetric signature. So with the crop stage also, this signature will vary. So this is very efficient tool for monitoring the crop condition, the crop growth, and so on and so forth. Now, another important crop is actually sugarcane. This is a high biomass crop. So C-band data sometimes is not sufficient to monitor till the end of the crop stage, till the grand growth and the maturity stage. So a combination of the Sentinel and L-band data, Sentinel C-band and ALOS Pulsar L-band data has been used to monitor this crop, where you can see the response of these various polarimetric parameters across the various stages like germination, tillering, grand growth and maturity has been monitored, and a good sensitivity has been observed. So there is a moderate sensitivity in C-band, which was actually improved when we used the L-band data. So this kind of studies actually for crop monitoring and parameter retrieval for especially the medium and high biomass crop like millets, maize, sugarcane, we may require little higher wavelength like S-band and L-band data apart from C-band. These are some of the findings. Thank you very much for your kind attention.